Hello, in this lecture, we're going to be talking about molecular genetics. So this will be an introduction to that. And then I just wanted to start this off with looking at a question of how we could possibly combine the DNA of 3 people. So when we look at where the DNA is, remember that some of it is in the mitochondria, but the vast majority of it is in the nucleus of the cell. So what we could do is we could take, if we were going to combine the genetic material, we would take an egg cell and we would remove the nucleus. Okay. We would then have the little mitochondria in there. So that would be the mitochondrial DNA of a particular person. We would then take an egg cell and we take the nucleus out. You can do this with a little micro pipette and put into the egg cell. And so this egg cell has had its nucleus removed, so they call it the enucleated egg cell. So essentially, this cell has become kind of a surrogate, and it's going to take the nucleus from another individual, and you're going to put the nucleus into the egg cell. And then finally, you're going to add the genetic material from the father. So this would be the sperm. So this would be like individual 1, this would be individual 2, and this would be individual 3. So you might want to do this, for example, if the female that wanted to get pregnant had mutated mitochondria. So if there was a bad mutation in her mitochondria, and that sometimes happens, then you would want the mitochondria from another female, and you want to put the nucleus of that female in because she doesn't have any mutations and she wants to be the main person to pass their genes on. So this is actually an example of how you could combine the DNA of 3 people. And then obviously this might raise some ethical questions with some people, whether or not this should be something that we do. Okay, so today we're going to start talking about molecular genetics. And so we need to go back to what the genome is. So the genome is all the DNA. So we talked about DNA because it's a nucleic acid. So it's all the DNA in a person. What's really interesting about this genome is it tends to be the same, and it is the same in all of your cells. Because you start out as a single cell, that cell then passes its genome on, it duplicates its genetic material, and then divides via mitosis, and it passes all its genetic information on to the next cell. Generally, the cytoplasm gets split up, and so the mitochondria are split up as well, so that the mitochondria that are passed from one generation to the next in a person's life within their cell cycle tends to be the same, okay? So when we do, and we look at the DNA in a person, we now estimate that this is about 3 billion, what we call base pairs. Okay, so we're going to talk about what this is, what a base pair is in a little bit. So that's a lot of DNA. The base pairs are given letters, so it'd be A, G, T, C. Those are the letters. So that would be like the genetic code. And it's the sequence in which those are put together that gives us the whole genome. So when we were sequencing the human genome, we were trying to figure out how those letters all went together and how some of the DNA codes for protein and other parts of the DNA do not code for protein. So we have lots of genetic information. So when we looked at the Human Genome Project, which was finished in the early 2000s, what we were trying to do is we were able to do is we were able to sequence the base pairs. And obviously we had to pick one person to do this with, but we kind of assume that that person is going to be pretty similar between people, specifically in really important sequences that code for important proteins. Although there is going to be variation between people, and so we don't really know about how important that variation. Well, we know variation is important, but we don't know, how important that is. But so the human genome has been sequenced, as have other genomes. So like the fruit fly, the Drosophila melanogaster fruit fly, they have been sequenced, and some other things like the nematode, the roundworm, which is called C. elegans, that has also been sequenced. And so our genome is not the only genome that we sequence. We also sequence other genomes to understand the pattern of genetics in those organisms as well. And also we have lots of genes that we share with other organisms, and that's significant. So when we were going about this, you have to remember that Mendel had no idea what the genetic material was. And neither did Morgan, who worked in the 1920s with fruit flies. So we're moving into the 1950s here, and that is where we finally figured out what the genetic material was and how it works. So when we were first starting to figure out what the genetic material must be capable of, so we were trying to figure out what the genetic material was, we had three things that it had to be capable of. So the first thing that has to be capable of is it must be capable of making copies of itself. The second thing that it must be capable of is coding for differences observed between individuals. Within a species, like different people, but also like between humans and earthworms. So coding for differences observed between individuals. And the third thing is it must capable of undergoing rare changes. So a good example of the rare changes would be an example of a mutation. And so mutations are random. So mutations are random changes in the sequence of nucleotides. Okay. It's important to realize that mutations are random. We do not mutate towards a goal. But once we have a mutation, we can have selection, and selection is non-random. Okay. When we talk about making copies of itself, remember that we talked about how chromosomes can go from being unreplicated, and then prior to cell division in the S stage of the cell cycle, it can be replicated. So we can go from unreplicated to replicated chromosomes. And then we're going to talk about what the genetic code is and how we can read it. So this would be really important to study. This would be a really good essay question to ask you, what must the genetic material be capable of? So you should memorize that. Okay, so let's look at the DNA itself. So the DNA is composed of nucleotides. And the nucleotides have a sugar. They have a phosphate group. And they have a nitrogenous base. So the nitrogenous bases are A, which stands for adenine, T, which stands for thymine, C, which stands for cytosine, and G, which stands for guanine. That's a G, guanine. Okay, So those are the nitrogenous bases, and that's the letters that we look at, okay? The other thing about the DNA is that it is double-stranded. And there are base pairing. So with base pairing. And so there was a scientist who figured out that when we looked at all of the bases in our DNA, we had the same amount of A's and T's and the same amount of C's and G's. And this is what is referred to as Chargaff's rule. So A, hydrogen bonds with T, that's a weak bond. And then C will hydrogen bond with G. Okay, so that is the double-stranded with base pairing. We also have what is referred to as a helix. And so this 3D structure of the DNA is called a double helix. And this idea of this model of what the DNA was published in 1953, and the scientists you might have heard of, which are James Watson and Francis Crick. And they actually wrote a book called The Double Helix, which is a little thin novel which describes all the cool stuff that was going on at the time when there was the race by many, many different scientists across the world to figure out what the genetic material was. And so they eventually won the Nobel Prize for providing their model. So when we look at the double helix, first, actually, let's look at the nucleotides. So you don't need to know these words, but I just wanted to point out that some of the nitrogenous bases are single-ringed, and they're called pyrimidines. And some are double-ringed, and they're called purines. Notice that they have nitrogen atoms in them. This is the nucleotide. So you should be able to identify the components of the nucleotide. So this is my nitrogenous base. I know that because it's got that N in there. This is my sugar, right? It's a 5-carbon ring sugar. It's ribose, or deoxyribose, actually. And this is my phosphate group. It's got a P. So if we look at how these are arranged, the sugar phosphate makes up the sides of the helix, and then the rungs of the helix actually are made up of those base pairs. So you'll notice here that the sides of the ladder would be the sugar phosphate groups. So this is the side of the ladder right there. These are the nitrogenous bases, so they make the rungs of the ladder. And notice that these hydrogen bonds are really weak. So when you apply heat or enzymes, you can easily separate the DNA molecule into 2 single strands. Sometimes this is called the sugar phosphate backbone, and then the bases are the rungs of the ladder. Okay. We're not going to worry in this class about 5' to 3', but they actually run in opposite directions to one another. So they're kind of anti-parallel. The helix is the way that it rotates. And so the rotation of the ladder is what makes up the helix structure. So if you think about it, we did not know the shape of the DNA. We knew what it was made up of, but it was 1953 when that was first published, the model of what DNA looks like. So this is just a picture. This would be the sugar phosphate backbone, and these are the base pairs. So when I say that there's 3 billion base pairs in our genome, that would mean that there are 6 billion nitrogenous bases in our genome, because if there's 3 billion pairs, this would be a pair, and then each one is a base. So there would be 6 billion bases in our genome. Okay, so if we look at how the DNA is replicated, it is said to be semi-conservative. So what that means is that the strands of DNA separate. So strands unwind and separate. This is due to an enzyme, and it's kind of easy to remember. It's called helicase. So let me put an I in there. So helicase is the enzyme that unwinds and separates the strands. Okay. One strand, which is called the parental strand, serves as a template for creating a new strand, which is called a daughter strand. It could be, I don't know why they use daughter, it could be son, but they tend to use daughter here, like daughter cells or sister chromatids, right? So they produce a daughter strand. Now, sometimes you can get mispairing, but pretty much the enzyme that is involved in creating the new strand is called DNA polymerase. And this is easy to remember because ACE is the enzyme, a polymer is a large molecule, and DNA. So DNA is just, this is the enzyme that matches the A's and the T's and the G's and the C's to produce a new strand, okay? So if we look at a diagram that shows this, these gray ones would be the parental strands. This is the new strand, and so A would pair with G, or excuse me, A pairs with T, G pairs with C. So if I give you on the quiz a strand, okay, so this is one strand of DNA. You could act as DNA polymerase and you could build me another one. So A, based upon Chargaff's rule, always pairs with T. G always pairs with C. A, C, A, C, T. Okay, so if given one strand, you should be able to give me the complementary strand of DNA molecule. And that is what is referred to as DNA replication. So this actually represents the DNA polymerase. This represents the daughter strands, and this actually represents a mistake. So notice that G pairs with T. So the other thing that happens during DNA replication is that we have what is called, and this is important because bacteria and mitochondria don't have this, but we have what are called proofreading enzymes. So they check the daughter strand for mistakes. So they check for replication mistakes. And they can cut those mistakes out and repair them. So this just shows they could repair that by cutting the T out and replacing it with a C. And they can also cut this one out. This is just where you have a covalent bond inappropriately between two T's. We'll talk about what this is. UV radiation can cause this. And the proofreading enzyme just cuts here and then reads it. And so this is important because this helps to prevent mutations. So this decreases the number of mutations that would arise normally. Some mutations get through the proofreading, but in other mutations do not. So a lot of mutations get proofread and then fixed. Okay, I'm going to skip the DNA testing. Okay, so I want to talk about how DNA codes for protein. So you'll remember that your DNA is located in the nucleus and it does not leave the nucleus. So in order to get that information out from the nucleus to the ribosome where proteins are produced, we have to make a copy of the information that is in the DNA molecule, okay? So I'm going to show DNA here is in my nucleus, And then I'm going to produce what is called a messenger RNA. This can be abbreviated as mRNA. This messenger RNA leaves the nucleus and it goes out to the cytoplasm and it binds to a ribosome. And then it is translated into protein. So this going from DNA to messenger RNA, this is called transcription. And the reason why it's called transcription is because we are just making a copy. So we're transcribing. So it'd be like going to the library and you have some information and you make a photocopy of it. It's in the same language, but you're photocopying it so you can take it out of the library because they won't let you take the book out, okay? And then here, this is called translation. So translation is where we take messenger RNA and we turn it into protein. These are two different types of language. Protein consists of amino acids. Messenger RNA consists of nucleotides. So we need to translate it. So think English to Spanish would be a translation. Nucleic acid language to protein is translation, okay? So we need to look at RNA. So RNA is similar to DNA, except for the fact that it is single-stranded. It has a different sugar, slightly different sugar. You don't need to know that. And instead of thymine, there is no thymine. So if you see a T, you know it's DNA. But instead, we have uracil. So uracil is another nitrogenous base that simply replaces thymine in the RNA. Okay. So let's look at transcription first. We're not going to talk about that. So let's look at transcription first. Yeah, we'll talk about that later. Okay, so let's say that I have a piece of DNA that I want to transcribe, and we only transcribe one strand. So I'm just going to kind of randomly put in some letters here. Okay. So notice here that this is the coding strand of the DNA. There would be another complementary strand here, right? And you could fill it in. So A would pair with T, G pairs with C, etc. Okay, but this is my coding strand of DNA. So I need to transcribe this. So I need a process called transcription. And the enzyme is very similar to DNA polymerase, but it's called RNA polymerase. So that is the enzyme that is involved, and it binds to specific sites on the DNA that give it a signal. It's a sequence of nucleotides that signal it and say, bind here if there's a gene present, okay? So the gene is turned on and it's gonna produce a protein. So A pairs with, in this particular instance, there's no T, so we go U. G pairs with C. C pairs with G. T pairs with A. T pairs with A. T pairs with A. C pairs with G. G. And G pairs with C. Okay? So notice that I still applied Chargaff's rule, but where there would have been a thymine, I use uracil. Okay, that's really important. Okay, so the messenger RNA, we're going to talk about it processing, but we're not going to talk about that today. So this is going to leave the nucleus, and there's little pores in the nuclear envelope that allow it to leave the nucleus and go out into the cytoplasm, and it is going to be translated. And this is where we need to know the genetic code. And that has been determined, and it is universal. And that's going to be really important when we talk about biotechnology, because all organisms use the same genetic code. So that means that if you put a plant gene into one of my cells, I would be able to read it and produce a plant protein. Okay, so this is using a table. Okay, so you need to know how to use the table. And so I have, probably have a table here, but let me get one up. So we have messenger RNA. You can just Google the genetic code, and you'll get pictures, you'll get images, and you'll get an image of a table. And I also went to Khan Academy. It sent me to Khan Academy. So I'm just going to put it here. Oops, I guess it's too big. I can't put it here. But this is what the genetic code looks like. So let's look at that first one, UCG. So UCG, UCG, that is, oops, that is serine. So this would be serine. So I can write that down. UCG is serine, okay? And then I look at the table again and I find AAA. So I look at the first letter, second letter, that is lysine. And then I go, so AAA is lysine, and then GGC, I look in my table and I go GGC, GGC, and that's glycine or GLY. So this is the sequence of amino acids. Okay, so I would use the table to determine that. So you should be able to, if I give you one strand of DNA, using Chargaff's rule, give me the other strand, and I'll tell you which strand to transcribe. So that's the coding strand. And then you're going to produce a messenger RNA, and the genetic material has the messenger RNA codons. So that's important. So this 3-letter is a codon. These 3 are another codon, and these 3 are another codon. So the genetic code is a triplet. It's a triplet code. And the reason why it has to be triplet is because we have about 20 amino acids. So we have 20 amino acids. And we have 4 nucleotides. So think about it. If it was a single letter, and each nucleotide, like U coded for an enzyme, U coded for, an amino acid, C coded for amino acid, G coded for amino acid, we could only code for 20 or 4 different amino acids, not 20, right? If we had a 2-letter code, that would still not give us enough combinations. So it has to be a 3-letter code, okay? Now, the other thing that's kind of interesting about this genetic code, which we'll get into in the lecture, is that there is punctuation. So these are what are called stop codons. You don't need to memorize them. They're in the table. This table will be given to you. This one is a start. So AUG, methionine, that is usually at the beginning of a gene that is going to be translated, or messenger RNA that's going to be translated, okay? So all of this translation takes place at the ribosome. And so we're going to talk about how it does that at the ribosome. And we'll talk more about this production of the messenger RNA. So this is just an overview of how the DNA codes for protein. So we'll talk more about it in the next lecture.